Thank you. After the last several talks, I think you're going to be hearing a similar refrain. As you can see, the title has slightly changed, but the overall impact will be the same. The main thing I'd like to emphasize, in addition to the cells, animal, and people of what we're doing, is that there are challenges which we're beginning to hear about. And I have to be able to work this, right? Okay. All right. My collaborators owe a great deal of thanks. At Downstate, Anna Miller has performed all of the in vitro experiments, Richard Feynman, of course, and the human studies have been done at Einstein and Montefiore. Newer data that we're hoping to obtain soon will require the expert assistance of Alex Perlman and EU Zoo. We've been funded, as previously in the past, through the Atkins Foundation and State University of New York Research Foundation, and recently through crowdfunding with experiment.com. That's a story of an entirely different nature, so I won't discuss that now, except to say that Pete Evans, who is a low-carb chef in Australia, and Tayo Prayasito, who is a low-carb chef in Indonesia, were particularly generous donors to experiment.com. So this is a perspective of our work and a prospective of what we plan to do, as I say, based on our experience and the work of many others, most of whom sit in this room. Cancer is described as a heterogeneous genetic disease, and with, I think, in agreement really with a somewhat moderated approach to Dr. Seyfried's, probably more in line with Dr. Scheck said. I think we have to think of it as a genetic and a metabolic disease which are intertwined. I really liked the model of the yarn, the spool of yarn, which was totally tangled, because they're both the metabolic and the genetic changes, I think, are going on more or less simultaneously. And the genetic ideas, however, and the molecular ideas, which we'll see in a minute, while likely having some validity, as I say, they invite very complex therapies which don't usually work too well and have pretty serious side effects. So we're all familiar with the traditional multiple cocktails of toxic chemo which then fail. And now there are emerging gene-targeting therapies, so-called personalized vaccines, and there are further emerging molecular targeting of important molecules, signaling molecules in cells which are responsible for growth and resistance to apoptosis, molecules such as mTOR and PI3 kinase and so forth. Now, as far as the emerging vaccines are concerned, these personalized vaccines are fairly impractical as far as I can tell. They're going to be hugely expensive, rather toxic and invasive. In order to get antibodies to all of the antigens involved in a very heterogeneous tumor, very heterogeneous cancer, you have to sample all the tumor sites. So if you have a metastatic disease, that's wildly impractical and extremely invasive, and you're still not going to succeed for the reasons that Dr. Scheck said. A cell over here in a big tumor is going to be different from 1 centimeter away. And so if you want to provoke a complete antibody response, you're not going to succeed. Even radiation therapy for provoking an immune response is quite interesting, but you have to locate all the sites anyway and still therefore irradiate possibly multiple sites throughout the body very impractically. Now, as far as emerging molecular targeting, these are drugs that have been targeted to some of the more prominent signaling molecules. There's sirolimus or sirolimus, I don't really know how it's pronounced. Rapamycin is easier. There are congeners, everolimus, and they inhibit mTOR, so they inhibit the growth signaling molecule. And there are others. I don't really want to go into them other than the fact that there are drugs for each one, and they all have side effects. Okay, metformin is pretty well tolerated, but even so, it will also have side effects. So let's just look at rapamycin just as an example. Now, the side effects of rapamycin include hyperlipidemia, basically type 2 diabetes, mucositis, and an impaired immune response. And generally speaking, these things are not very good for people who have cancer. And I just quote from an article in OncoTarget, an editorial written by Dr. Solomon, called About Face on the Metabolic Side Effects of Rapamycin, in which he ends up saying that these side effects could possibly be alleviated by pairing rapamycin with rosiglitazone. Well, isn't that nice? So are we going to now introduce yet further drugs to counteract the side effects of the existing drugs, which we didn't like to begin with? So, I think we're missing something, and I think people in this room know exactly what we're missing. So rather than targeting complex protein or gene expression with vaccines or with toxic drugs, let's take advantage of the commonality of most advanced cancers. And we know what this is. Most cancers display the glucose glycolytic dependent phenotype, the so-called Warburg effect, as beautifully described, and as this is often evidenced on a positive PET scan, which is then potentially responsible to ketosis, as we'll get to in a minute. Let's just move quickly to the next slide. This is coming out very poorly. Is this contrast as bad as it looks to you? Well, anyway, this is supposed to show distribution of FDG, fluorodeoxyglucose. You can see that there's glucose in this radioactive compound. I'm sorry for the quality of the scan. The brain takes up in a normal patient, as often does the heart, depending on diet. There's excretion in the kidneys and the bladder. But this is a metastasis in the liver in this patient, and you can see that after failed chemotherapy, you have the same metastasis with additional new ones in the liver, and you can't see the 2 additional ones that are also in the lung. Okay? But a PET scan is basically an in vivo image, which is an explicit manifestation of the Warburg effect. It's actually how I got interested in the field. So most cancers display this glucose and glycolytic dependence, and that's one of the commonalities. And the second is there are common growth-promoting apoptosis-resisting signaling pathways which are downstream of the insulin receptor. So let's take a look at that. The relevant molecules downstream of the insulin receptor on cancer cells therefore are inhibited by insulin. So that reducing insulin signaling, in fact, hits all of these targets simultaneously and without side effects, because we know that inhibiting insulin through diet has minimal side effects. And they look pretty familiar because they're the same targets that I showed you a couple of slides ago that had all those drugs targeting it. So it seems that ketogenic diets might have value in inhibiting insulin. And this is, I like to put this because this is a drastically oversimplified signaling molecule diagram, which doesn't look drastically oversimplified to me, but apparently it is. And on it, you can see that here are the signaling molecules. There's PI3 kinase, mTOR, AKT, there's AMP kinase, all the things that I mentioned on the prior slide as all drug targets. There are also some other ones. There's the RAS, RAF, MEK, ERK pathway. I always say that when I first was exposed to this, it sounded like alphabet soup to me. I'm getting a little bit more used to it, but it still sounds that way a little bit. But anyway, I guess the point of the diagram, however, is that, of course, upstream of all of these molecules is the insulin receptor. There it is. And if you inhibit the insulin receptor, it turns out you affect all of those molecules exactly in the direction that you want to, and you haven't given a drug. So that's pretty good. So ketogenic diets result in maximum inhibition of insulin. The insulin receptor is on the cell membrane. So to repeat what we've been repeating basically all conference now for cancer is why not inhibit insulin secretion to inhibit all the signaling molecules simultaneously to reduce growth and proliferation, which some of those molecules stimulate, and to reduce the resistance to apoptosis, which they also, and those are some of the hallmarks of cancer. And the second point, which is that you're not only reducing insulin in a ketogenic diet, you're producing ketone bodies. And so the question is, can ketone bodies themselves inhibit cancers? And we have evidence that they do, and we have conflicting evidence as well, but let's take a look at some of it. And they can inhibit cancers in a variety of ways. A little faster than I wanted it to show, but first is something you've heard about already, the fact that ketone bodies have been shown to act as histone deacetylase inhibitors. And this has been shown by a variety of investigators over the last couple of years, and histone deacetylase inhibitors are being used in cancer therapy now because they themselves can cause apoptosis of cancer cells, and or they can inhibit proliferation and growth of cancer cells, or both. They may do both. Second, we've spent some time showing in a particular group of experiments in vitro direct inhibition of ATP production in several lines of cancer cells not seen in control fibroblasts. And this inhibition of ATP production, since cell growth depends on ATP, also results in growth and proliferation inhibition. And finally, we've heard from Dr. Boros that mitochondrial matrix water stabilization by ketone bodies will stabilize mitochondrial matrix water compared with what glucose does, and that this can also inhibit ATP production in mitochondria. Now, as far as HDAC inhibition, the brief description of it is that HDAC inhibitors disrupt the normal epigenetic regulation of gene expression. Again, some beautiful slides that Dr. Sheck showed. And by altering gene expression, this can result in reduced cell proliferation, increased apoptosis, or both. Furthermore, it's been shown that short-chain fatty acids, including butyrate, valproate, hydroxybutyrate, and acetoacetate, have HDAC inhibitor activity. Now, beta-hydroxybutyrate and acetoacetate act as anti-cancer drugs do, but without toxicity, and you don't have to administer them, except you could if you wish, but you could also put a person on a diet. So, John Mary Addison performed some very elegant experiments showing some HDAC inhibitor effects using butyrate, and in his experiments, he tested, it looks like, about 30 different colon cancer lines, which was his interest. And in comparison to the gray bars, which are the gray bars here are carrier material, you can see that 5 millimeters of butyrate had about, in these cases, maybe tenfold as much apoptosis as did the carrier material, the controls. And about 2/3 of these cancer lines had truly significant apoptosis as a result of incubation with 5 millimolar of butyrate. Furthermore, he also showed that that cancer lines that were sensitive to this also showed inhibition of proliferation. And Shimazdu, and Newman also showed that beta-hydroxybutyrate particularly, and also acetoacetate, both acted as HDAC inhibitors. And so this has not been explicitly tested by Mary Addison in these cancer lines, but in fact we can anticipate that possibility. Second, I'd like to talk about the direct metabolic inhibition of ATP production in cancer cells due to overproduction in cancer cells of UCP2. I went into this a little bit last year, and has to do with the fact that most cancer cells really are living on the edge of death due to overproduction of reactive oxygen species. And uncoupling protein 2 has the effect in cancer cells of just managing those ROS at barely survivable levels. ROS is in some ways the dollar bill of the cancer cell. It both provokes the cancer cell to mutate towards a more aggressive form, or it kills it, and the cancer cell becomes a suicide warrior on behalf of the team. But it doesn't matter because the next cell will go on and do something bad. So overproduction of UCP2 is in fact quite common to virtually all cancer cells that I've seen. And it has a secondary effect, however. Well, first of all, because it's an uncoupler, as it happens, there's an effect in which uncoupling protein 2 acts as an inefficiency protein. And in fact, what it does is it causes reduced efficiency of production of ATP in cancer cells when you incubate them with beta-hydroxybutyrate and acetoacetate. Actually, acetoacetate, I should say. And so this is the experiment that we did, in which we compared 3 control fibroblast lines with 7 cancer lines, 2 breast and 5 colorectal. And it's a very simple experimental design. The hypothesis is basically that we look at, let's say this is a Petri dish, and you have a certain amount of cells in the Petri dish. At 4 days after glucose medium, you get this density of cells, which we'll call 100%. And in cancer lines, you do the same thing. This is your baseline density, and this is your 100% at 4 days. But if you grow the cancer cells in glucose medium plus acetoacetate, a ketone body, we're expecting the control fibroblasts to have no growth inhibition, and we're expecting the cancer lines to have growth inhibition. And rather astonishingly, that's exactly what we found. And so this was all 3 cancer lines. We're showing the 100% bar is the black bar here. And you can see that that means 100% of blue is growth and red is ATP. And all 3 fibroblast lines show 100% of growth and 100% of ATP at 4 days of incubation, whereas the cancer lines all show a different degree, however, of both inhibition of growth and ATP at 4 days, depending on the line. The least seen in MCF7, a breast cancer line, and the most seen about 50% of growth and ATP inhibition in one of the colon cancer lines. And in fact, just from that particular slide alone, I think you can appreciate exactly how amazingly linear the relationship is of the growth to ATP. They line up almost perfectly, the relationship of growth to ATP reduction, and that's what we see. It's a little bit unusual in biological experiments to see an R value of 0.948, where in fact you see that the controls lie on the same line as the cancers because they have 100% of growth and ATP, whereas the cancers have a lower amount. But the R value is 0.95, and it's very tightly correlated. And sure enough, the cancers all overexpress UCP2 in comparison to the controls. And in fact, as we would also expect, you can see that the least UCP2 production correlates with very well with the most ATP, and the more UCP2 you have, the less ATP is produced. So this was shown in cell culture. So yeah, we just showed that. So others at this conference also have reported cancer inhibitory effects at the cell level or in animal models. Dr. Seyfried has shown this, Drs. Poff and D'Agostino has just discussed, Drs. Clement, Dr. Champ, Dr. Scheck, Dr. Boris and all. And I just want to acknowledge them because this is all essential work that's being done. Human trials have been mostly case reports or trials with small numbers and without controls. So this is a very significant limitation of what we're dealing with. They've been done by a variety of investigators over the years, some of which we've all mentioned already. Our study was basically a safety and feasibility trial of a low-carb ketogenic diet targeted to about 5% of total calories for 28 days in patients with advanced PET-positive cancers. And we did an entry and exit PET scan as a surrogate marker of efficacy, looking at the change in the FDG uptake as a surrogate marker. The patients were monitored for compliance as well as for the systemic metabolic effect of whatever compliance they actually achieved by a weekly fasting beta-hydroxybutyrate concentration in the blood. Dietary nutrition recalls aren't that accurate, so the beta-hydroxybutyrate was essential. Now, the baseline data showed here that you can see that the patients ranged in age from about 52 to 73. There were 7 women, 3 men. And most important, these patients were not picked by tissue type. You can see we had a variety of cancer types. They were picked by phenotype. That's to say their metabolic behavior. They were picked because they all had a positive PET scan. So this was a very broad study. It was recommended against that we did this, but in a way I'm glad we did because it shows the commonality of the process, namely the Warburg effect. The diets were all safe. None of these patients who stopped a day or two short stopped because of any unsafe adverse effect of the diet. One patient had a dental abscess, needed an extraction, and had to go on soft foods, and she couldn't stay on the diet. Another patient had plane tickets that she had reserved with advanced cancer, thought this was the last vacation she was going to take. We weren't going to stop her. These were not unsafe effects. So what you do see, one patient is a little different, biologically different from the others. All of the patients had multiple chemos except her. She refused everything. She had no surgery, no chemo, no radiation therapy, yet she was still alive 14 years later, and she had a positive PET scan, and we know this was an aggressive cancer. We had pictures in her file from 5 years earlier which showed chest wall invasion at that time. So, beggars can't be choosers. It took us 4 years to collect these 10 patients, and we didn't think of it at the time, but the fact that she was going to end up having stable disease meant we had to exclude her from analysis because she was growing obviously so slowly that we wouldn't detect her in a 1-month study as having progressive disease. So anyway, but the short story is that all the patients were calorie deficient for reasons that we tried to overfeed them. This was an outpatient study. Their dietary recall records suggested they ate 1/3 less on the diet than their predicted energy needs, and they all lost weight. However, the calorie restriction that they showed and the weight loss that they showed had no correlation with the outcome. That's interesting because we say that calorie restriction is important. It did not appear to have a relationship in this very small study. We can see that this is the segregation of the patients according to their metabolic response. This is the patient we had to exclude because she was too indolent. Her disease was growing too slowly. But you can see that the patients who had stable disease or partial remission was one of them, had ketosis of about 10 to 20-fold over baseline, and the patients with progressive disease had basically less than 5-fold baseline, but there was one overlap. Okay. And overall, there's no statistics here to speak of in a 10-patient study, but basically the patients with progressive disease partial remission had about 3-fold the level of ketosis if you want to make averages of these numbers. And as expected, ketosis and insulin were inversely correlated. So this was a prospectively designed and executed pilot study. 10 patients completed it without unsafe adverse effects. Ketosis was observed and it correlated with insulin inhibition. The extent of ketosis correlated with disease stability, and there was unplanned calorie reduction and weight loss, but this was uncorrelated with disease stability. But we have to be very careful about interpreting everything that we've talked about here. In vitro findings don't often extrapolate to in vivo results, including our own. Rodents aren't usually good models of human metabolism. We're all forced to use them, but we don't like them. The few human studies that have been performed have been in small numbers of patients and without control groups, including our own. And other caveats have to be considered, and let's just start with genetic mutations. Okay, we had this thing before. Okay, this was our signaling molecule diagram. If you inhibit insulin, it won't make any difference if you have a constitutive activation in the PI3 kinase molecule. That was shown by Kalani and Sabatini, that you will not affect downstream signaling molecules if you have constitutive upregulation of PI3 kinase or mTOR of any of those molecules. So the signaling molecules will get in the way if they have, and so you will then end up not affecting things like growth or inhibiting apoptosis. Okay? So that reducing insulin may not benefit cancer which have mutations in critical pathways. And I just want to point this out, that breast cancer has Stemke and Hale showed that there's a PI3K constitutive activation mutation in 35% of ER-positive breast cancer and 23% of HER2 positives. Endometrial cancers have about 50% mutations in that pathway. Colorectal cancers have about 40% in KRAS and 40% in PI3K constitutive activation. So this is a problem. However, cancers without such mutations still represent 65% of ER-positive, 50% of endometrial, and 35% of colorectal, right? And ketosis, which is due to liver function, right, which is independent of the cancer cell signaling stream downstream of the insulin receptor on the cancer membrane, therefore has independent effects that still might be beneficial. So you have two mechanisms in parallel. One is directly on the cancer cell from insulin, and the other is ketosis, which happens in the liver. So, but caveat two, ketosis may not always inhibit cancer. Mary Addison showed that the HDAC inhibition is present in most, but not all colon cancers tested. Our data were in vitro and relate only to 7 tested cancer lines. Glutamine is often an alternate substrate for aggressive cancers. That's a very interesting thing which nobody I've heard discuss much here, but I just want to point out that glutamine is regulated by one of a gene signaling protein which I didn't mention, MYC. And MYC tends not to be mutated, but it tends to be multiply expressed. You tend to get multiple copies of MYC, which then overregulates glutamine expression. Fascinatingly, however, HDAC inhibitors also may inhibit MYC, so we may end up getting that effect anyway. Well, anyway, we don't know, but it's all unproven yet in cancer, so a lot of work still has to be done. And finally, Michael Lasante's work, which was quoted, the so-called reverse Warburg effect, and others have also claimed that some breast cancer types actually feed on ketone bodies. Okay. So, finally, caveat 3, are all patients susceptible? We have to understand not just the behavior of the cancers, but also of the patients. And we would think that ketogenic diets to treat cancer patients, for example, with type 2 diabetes or with prediabetes, would treat both at the same time. This, however, has to be proven. It hasn't been shown. Insulin resistance is usually at the fat, muscle, and liver cellular level. What happens at the cancer level is as yet unknown. What happens when you inhibit insulin, it's not known what happens to the response of the GLUT receptors and things that may be at that. So this is unstudied. And in fact, in our recharge trial of, again, only 10 patients, the 4 patients who had progressive disease had the greatest evidence for prediabetes. So who knows? Anyway, none of the caveats and limitations should distract us from the potential advantages of a free and non-toxic metabolic approach in susceptible patients and cancers. That's the positive message. I want to come out with something positive here, and it's real. So our goals should be very simple. We need to identify the cancers and the patients in advance who are sensitive to insulin inhibition and or ketosis. So new directions that we've been trying to pursue is to expand up from a small study of 10 patients and to include controls. So right now we're working on obtaining funding for this. Fingers are crossed, but we'll see what happens. And so we're trying to scale up to 45 patients on a ketogenic diet and 20 controls on an isocaloric low-fat diet. And I won't go into the details because I think time is running short. We're also trying to couple diet and drugs. We want to study diet and rapamycin in a mouse model where we do 4 arms, standard diet plus rapamycin, ketogenic diet plus rapamycin, and we're going to measure a variety of parameters. And finally, we'd like additional ways of measuring ketone bodies in vivo, and we're in the process of developing a radio-labeled ketone body in conjunction with collaborators at University of Alberta. Stephanie Mattingly has been working on this and has made astonishing progress. She now has 2 precursors, which basically are at the point where she's ready to substitute directly for a fluorine 18 labeled beta-hydroxybutyrate and a fluorine 18 labeled acetoacetate. And these could then be used in humans. And I mean, well, not immediately. There would be an awful lot of work that would have to be done. I can't predict for you right now what exact value would come from this tracer because a lot of studies would have to be done. You'd have to do normative findings first, then you'd have to do it under different dietary conditions for cancers and for a variety of cancers and so forth. It would be the 3rd F18 labeled metabolic tracer after FDG and F18 glutamine. So as I say, its practical uses aren't explicitly known, but it may permit exploration of in vivo mechanisms of ketogenic diets in cancer and identification of susceptible patients. And so, you know, this is a terrible slide, but we're hoping it would look something like this, but better. And I want to finish with a quotation from James Watson, because obviously co-discoverer of the gene structure. This is from Cancer World and reported by Anna Wagstaff. The inherently conservative nature of cancer research establishment is the biggest obstacle today to moving forward effectively toward a true war against cancer because they're still too closely wedded to moving forward with cocktails of drugs targeted against growth-promoting molecules. So I find that remarkable coming from James Watson, who also says that he would, if he were in charge, he'd attack the uniqueness of the biochemistry of cancer cells. We still don't know the reason for the Warburg effect. You have to unleash biochemistry, and there's no biochemistry left because everybody moved into DNA. Well, not everyone in this room. So, thank you.